Hello, Medicos. Good day to all of you. This is Dr. BK for you, and today we are actually starting with a new region, that is the neuroanatomy or the brain. So, in this lecture, I will be discussing about certain basic concepts and parts of the brain. Followed by that, we will be discussing about the meninges. Now, our learning objectives will be today is mainly a short introduction about the nervous system. So, followed by that, I will be discussing about the subdivisions of the nervous system, parts of the brain, and then the meninges in detail. So, that is the three meninges, and we will also concentrate on the subarachnoid cisterns and how the CSF is circulated in a brief manner. Now, first of all, what is the function of nervous system? You all know our body is mainly made up of four types of tissues. Epithelial tissue, which is for the protection, and then you have the connective tissue. Okay, so the connective tissue mainly makes up for the your blood vessels, ligaments, areolar tissue, even your bones, skeletal system. Then you have the muscular tissue, which is of contractile in nature. So through its contraction, it can bring about various movements. And finally, what we have is a more specialized form of tissue, which is called as the nervous tissue. So the main speciality of this nervous tissue is it is mainly made up of neurons. So the neurons are nothing but the nerve cells. Now, what is the main function of the neurons? Okay, so it can conduct stimuli or it can carry information. Okay, so that information is actually processed mainly in your central nervous system. Okay, so the perception and then integration of the information which is received, the information which we receive is actually called as the sensory information. So that processed information, what happens, it is stored for also future reference. That is one thing. Apart from that, it also sends immediate commands or a response, and that is actually traveled again from the central nervous system to the peripheral nervous system through the muscles. Not only the muscles, even the other parts like your viscera and then your glands, blood vessels, everything, to actually bring about the desired movement or the reaction. So receiving the stimulus from the external environment, mainly the external environment, you receive information like the pain, temperature, touch, pressure. So these type, they are actually called as the exteroceptive sensations, and also proprioceptive sensations. So this proprioceptive sensations is mainly from your muscle. Okay, so mainly exteroceptive information and then your proprioceptive information from the muscle and joint, that forms your somatosensory nerves. So soma means a body or body wall, we can consider here. Okay, and it also receives information from your internal aspect, interior of your body. This is actually visceral sensory nerves, that is sensations from your viscera. So like your gastrointestinal tract or your respiratory system, your cardiovascular system, or excretory reproductive system, so they also receive stimulus. Most commonly you have visceral pain. So then Followed by that, you also have whether, for example, your stomach is full, so your bladder is full. So these are also visceroceptive uh, sensations. Okay, so that is also what happens is that information is also processed, and then what happens, it gives appropriate commands even to the viscera, which is called through the visceromotor nerves. So the visceromotor nerves are again part of your sympathetic and parasympathetic autonomic nervous system, because it is not under our control. So this visceromotor mainly, it innervates your glands, smooth muscles. So smooth muscle means you should always remember, it is not only the smooth muscle which you are seeing in the gut, inner circular or outer longitudinal. The smooth muscle is actually present in almost all the viscera. So for example, your uterus has got a smooth muscle, uterine musculature, myometrium. Your bladder has got a smooth muscle. There is smooth muscle in your bronchi. Okay, so like that in all the visceras, you have the smooth muscle, and especially also your cardiac muscle, which is also under the control of this autonomic nervous system. Okay, so the stimulus actually is processed, and then you react to the stimulus through appropriate action. So this diagram, what you are able to see, also shows a reflex arc. So you receive sensations from this finger, whether something hot or somebody pricks it with a pin, or you come in contact with any other surface. So naturally, based on the information receives, what happens is some action has to be taken to this part of the body. So whether if it is a undesired sensation, naturally you have to move away from that sensation, or mostly it is protective in nature. Now, a simple reflex arc consists of an afferent neuron. You are able to understand, and then what happens is uh, it has the sensory receiving an afferent neuron, then you have an efferent neuron, and then the effector organ is your muscle mostly. Okay, so this is called as a simple reflex arc or a monosynaptic reflex. Sometimes you are able to see an interneuron is interposed between the afferent and the efferent new nerve fiber. Then it is called as a polysynaptic reflex. Okay, so these interneurons, mainly what happens is they are capable of transforming the information to even the opposite side of the body. They might connect to the opposite side neuron, or they might actually, what happens is, transfer this effect to the other segment. So this especially happens in the spinal cord. Now, for example, if the information is received to from the particular segment of the spinal cord, so that effect has also is transferred to upper or lower segments. More than the received segments, it is also spreads to the other segments to bring about the desired movement. That is one thing. Second thing is, numerous afferent neurons, what happens is, might actually come and end in one interneuron. So this is actually called as the divergence. Or sometimes from what happens is, from interneurons, the effect, what happens is, might go to different efferent neurons. Okay, that is actually called as the divergence. So one is the convergence, another one is the divergence. So most of these aspects I have discussed in the general anatomy. Okay, so in the general anatomy lecture, kindly refer to it in the nervous system. So in the nervous system class of the general anatomy, I have discussed in detail about this reflex arc, and what is a typical spinal nerve, what are the types of neurons, types of axons, how do you classify them, and what is the synapse, structure of the synapse, everything. Please refer to the general anatomy class. So here your central nervous system processes the information which you are able to see here. General sensations from the skin which we have seen. Same way it also receives special sensations from your special sensory organs, like for example eye it is shown. You also have ear, then you have the tongue, okay, nose and all. Okay, so visceral sensations, as I told you, sensations from the viscera. Then the motor component, mainly it actually supplies the muscle to bring about the desired movement. Then, as I told you, visceromotor to the cardiac muscle. So this is very specialized type of muscle. And as I told you, it is through the parasympathetic component. It is also innervating the bladder, most of the viscera, except the heart. The heart is a special type of muscle, but here you have the smooth muscles. Okay, so somatomotor is mainly to the muscles and to the glands, blood vessels, and the to the viscera it is called as the visceromotor. So it also acts in concert with the endocrine system. So the endocrine system secretions and the nervous system acts together in maintaining the internal environment, which is actually called as the homeostasis. So whatever the external environment, whether it is a cold climate or an hot climate, we maintain an internal environment which should be always constant. Now, for example, you know our body temperature should be or should not exceed more than the 98.4 degree Fahrenheit. Then we call it as fever. So our body adjusts mainly, not adjusting, it actually takes part in various mechanisms in maintaining the internal environment. For example, in a very hot climate, what happens is you sweat a lot. Are you able to understand? So this is one of the mechanisms which is called as the homeostasis. So coming to the subdivisions of the nervous system, uh, the nervous system is broadly classified into the central nervous system and the peripheral nervous system. So the central nervous system mainly constitutes your brain and the spinal cord. Okay, so you are able to see the base of the brain, the cerebrum and the cerebellum and the brain stem, and it continues down as the spinal cord. So a thick long neural tube or a nervous tube, okay, spinal cord, which is approximately 45 centimeters in length. Now the peripheral nervous system is actually uh, serves as a bridge between your brain and or the central nervous system and the external environment. Not only the external environment but also bridge to the internal environment. So the main function of the peripheral nervous system, which consists of your cranial and the spinal nerves, which consists of your cranial and the spinal nerves, they conduct or receive the information. Okay, so they receive the information and then carry back the information which is sent by the brain. So that is the main function of this peripheral and nervous system, which consists of cranial nerves and spinal nerves. So cranial nerves, 12 pairs of the cranial nerves we have. Okay, so these 12 pairs of the cranial nerves, most of the nerves we would have discussed in the head and neck uh, topics. So in the orbit, third, fourth and the sixth nerve we would have discussed. Okay, trigeminal nerve extensively we would have been discussed in the infratemporal fossa and also in the orbit. Okay, so abductor nerve again in the orbit we would have discussed. Facial nerve in the face and also in the parasympathetic ganglia and in the ear class, part of the facial nerve. Vestibulocochlear nerve in the internal ear we would have discussed. Okay, same way ninth nerve with respect to the parotid gland, what it supplies, and also one muscle in the pharynx. So 10th nerve we have discussed in various regions. So in the neck to some extent, and then in the abdomen and thorax. So the 11th nerve, accessory nerve, again we would have discussed in the pharynx because it innervates the muscles of the pharynx, and also two muscles in the neck, especially in the posterior triangle. And the hypoglossal nerve also we would have discussed in the tongue region, and also in the digastric and submandibular regions. So only bits and parts here and there, mainly their supply, area of supply, we would have discussed. Now in the coming classes, especially in another 2 or 3 classes, we will be discussing the nuclei of this cranial nerve, which is mostly situated in the brainstem. Okay, so only thing what you have to do is you have to integrate this, and you will get the whole picture of these cranial nerves. Okay, the peripheral nerves, you are able to see 31 pairs of the peripheral nerves, 8 cervical, 12 thoracic, and then 5 lumbar, 5 sacral, and finally you have 1 coccygeal nerves. So they, what do they innervate and their functions, they are actually given here. So mainly the upper cervical nerves, 2 to 3 for the head and neck muscles mainly they innervate, and scalp, and from their diaphragm, and then slowly what happens, it innervates the lower cervical, is taking part in the brachial plexus. Then thoracic nerve, intercostal nerves mainly innervate your chest wall, muscles of the chest wall. Then as you go to the lower thoracic nerves, they are also lower 5 nerves, they are also innervating your abdominal wall muscles. Then lumbar nerves, they are mainly again taking part in the lumbar plexus, which is innervating your lower limb. Okay, and then from here lower lumbar and sacral nerves are innervating your pelvic organs. Okay, so this is a very superficial explanation of these, and at the appropriate regions we have also discussed about the spinal nerves. So 12 pairs of cranial nerves and 31 pairs of the spinal nerves. Then one more part of the nervous system, which is an offshoot mainly from the peripheral system, okay, is your autonomic nervous system. So it consists of the parasympathetic and the sympathetic components. So again in detail about this in the general anatomy lecture of the nervous system I have discussed. So both have contrasting actions, the sympathetic and the parasympathetic nervous system. Okay, so for example, you take parasympathetic is actually slowing your heartbeat, whereas in the sympathetic fibers increase your heartbeat. So these are all based on the situation it acts. Okay, this is actually energy conserving part of the nervous system, autonomic nervous system, rest and digest, whereas this is sympathetic, mainly energy expenditure. It comes in fight or flight or fright, when the body is senses something wrong or danger. Okay, so whether it might be structurally within the body or sometimes even emotionally, if you are feeling what do you call in some amount of restlessness. So here you are able to see the sympathetic chain, a series of ganglia starting from your cervical. It goes on up till the end. You have cervical part, thoracic, lumbar, and the sacral part. So this is again connected to your peripheral nerves, that is the spinal nerves, mainly through the gray and white rami communicants. So not only it is connected to the spinal nerves, also to the cranial nerves. The fibers pass via the blood vessels and they actually innervate your head and neck. They pass via the branches of the cranial nerves. So basic structure of the brain, anywhere if you take a section of the brain and if you see that it is mainly made up of this outer gray matter and the inner white matter. Okay, so mainly it consists of the outer gray matter and then the inner white matter. Okay, but you look at the spinal cord, it is quite opposite. It is reversed, where you see the inner gray matter and outer white matter. Okay, so this is mainly that is how the topography or the structures are arranged, uh, mainly for the functional reasons. So as you go along the structure of the various parts of the brain one by one, then you will be able to understand why this type of arrangement is present in the brain. Because very one simple explanation is, your brain or the cerebrum is the highest seat of control. So the neurons are mainly in the gray matter which is present. Okay, now this has to receive the information through the axons which forms the white matter and send the information again through the axons which is white matter. So all the white matter, what happens is, as it travels down through the brain, it is in the periphery, because again what happens, the spinal cord receives the information and sends information through the spinal nerves which has to leave the spinal cord. Okay, and these spinal nerves, they have to enter via this gray matter. Okay, so they have to enter via the gray matter and then they have to ascend up. So that is one of the simple explanation why the outer gray matter and inner white matter in the brain, and in the spinal cord inner gray matter and outer white matter. So as I told you, the gray matter mainly consists of the neurons. Here when I refer to neurons, it is the cell body and dendrites and initial part of the axons are present in the gray matter, because they have to start from the gray matter. These axons which is present in the gray matter are unmyelinated axons. Okay, then the white matter, what you see is the myelinated axons. Okay, so this myelin is mainly a thick lipid sheath. So lipid is made up of fat, when you call it as in a very simple lame term. So this lipid sheath, what happens, when the light falls on the lipid sheet, it is white in color, and that is why it is actually called as the white matter. Okay, so white matter are mainly made up of myelinated axons, and in the central nervous system, the myelination is actually taken care of by the oligodendrocytes. Okay, so they are all glial cells, neuroglia, oligodendrocytes. Okay, so again about this myelination, glial sheets, the structure of the myelin sheath in the peripheral nerve, it is called as the squam cell. So all those things, please refer again. I'm telling to the nervous system of the general anatomy series. Okay. Now, apart from the gray matter which you see here, there are also buried deep within the brain substance is mass of gray matters. Okay, within the white matter you can see mass of gray matters. They, you call it as the nuclei. So deep cerebellar nuclei there, deep cerebral nuclei, cerebellar nuclei. You have, you also have nuclei within the brain stem. So many nuclei, especially your cranial nerve nuclei. So you call it as a nuclei. But whereas when you see a collection of neurons outside the central nervous system, it is called as the ganglia. So one ganglia, trigeminal ganglia, and the one more ganglia is the dorsal root ganglia. Each spinal nerve possesses a ganglia on the dorsal root, which is called as the dorsal root ganglia. So both are actually sensory ganglia which are made up of unipolar neurons, because the term pseudo unipolar nowadays it is not used, as it creates some confusion. So it is the unipolar neurons. Okay, so as I told you, in the spinal cord you have the outer white matter and the inner gray matter. So a photograph of the white matter which you are able to see here. So they are actually uh, arising from various parts of your lobes of the cerebrum, and finally they converge and then they pass to the brainstem, and from there to the spinal cord. Of course, they also pass to your cerebellum, which is present here behind and below a cerebrum. Okay, so I'm not going into details of these white matter as of now. Now, not only that, with respect to the brain structure, you have gray matter, white matter, and a nuclei. There are also some spaces or cavities within the brain. Okay, so in addition to the neurons and the supporting cells, the neuroglia and the blood vessels, you have some cavities within the brain. They are called as the ventricles. Okay, so the ventricles in the brain, you call it as ventricles, but in the spinal cord, within the center of the nervous tissue of the spinal cord, there is a long canal running throughout the length of the spinal cord, which is called as the central canal. Okay, now these cavities are actually lined by a specialized type of the supporting cells, which are called as the ependymal cells. Okay, so these ependymal cells are responsible for secreting the CSF. So CSF, what happens is, from the capillary blood, the CSF is actually filtered and it is produced or secreted into these ventricles. Okay, so they secrete the CSF, as I told you. Now let us try to understand the parts of the brain. So basically, I told you the nervous system is divided into a central and a peripheral nervous system. So peripheral nervous system consists of your somatic nervous system, which cranial nerves and spinal nerves, and the autonomic nervous system with the sympathetic and the parasympathetic component. The central nervous system mainly consists of the brain and the spinal cord. Okay, so the brain and spinal cord, how they are developed. So initially it is a long tube which is called as the neural tube. This neural tube, the upper part shows some dilatations which are called as the brain vesicles or primitive brain vesicles. That is the prosencephalon, mesencephalon, and the diencephalon. Okay, so when the upper part, what happens, shows the dilatations or enlargement called as the prosencephalon, mesencephalon, and rhombencephalon. The lower part of the neural tube remains narrow, and that forms your spinal cord. Okay, so the spinal cord is the remaining tubular part of the 3/4 of the neural tube. So the upper part has actually bulged to form the brain vesicles, and various parts of the brain are derived, and the spinal cord remains tubular and narrow. Okay, now coming to the brain, you have forebrain from the frozen cephalon, which develops into forebrain. Okay, then you have the midbrain, and then they have the hindbrain. So mesencephalon and rhombencephalon. Now coming to the parts of the forebrain, this prosencephalon or forebrain gives rise to telencephalon, which is your cerebral hemisphere. Okay, your cerebral cortex. And basal ganglia, hippocampus, all these things you need not worry about now. So these are all present deep within the cerebral cortex only. Okay, then I told you the forebrain or prosencephalon divides into telen, and other one is the diencephalon. So here you're able to see a mass of structure. Okay, this is called as the diencephalon, which constitutes your thalamus and the hypothalamus. So it is mainly a sensory integrating center. Okay, so the sensory, mostly it is sensory, 90% of it is actually sensory integrating center, the thalamus and the hypothalamus. So your prosencephalon can be divided into telen and diencephalon. So telencephalon is your cerebral cortex, diencephalon is your thalamus and hypothalamus. The midbrain is from the mesencephalon, it remains as midbrain only. Okay, so again need not worry about these two, what is the tectum and tegmentum. Okay, so midbrain, it remains, it is a singular structure. Then hindbrain is actually divided into metencephalon and myencephalon. So metencephalon is your cerebellum and your pons, and myelencephalon is your medulla oblongata, which continues below the foramen magnum as the spinal cord. Okay, So your hindbrain or rhombencephalon constitutes your pons, medulla and cerebellum. Pons, medulla and cerebellum forms your hindbrain. Now here I would also like to introduce one more term. What is the brain stem? Brain stem constitutes your medulla, pons and midbrain. Okay, so please note the difference between the hindbrain and brain stem. Hindbrain is developed from the hindbrain vesicle, the rhombencephalon, which constitutes your cerebellum, pons and medulla, whereas brain stem is not developmentally, it is categorized, so it considered as the stem which holds these large cerebellum and cerebellum. Okay, and that is why it is made up of the midbrain, pons and medulla is the components of the brain stem. Okay, then as I told you, each of the brain vesicle, so prosencephalon has got telan and diencephalon, has got a cavity. The telencephalon is the cavity within the cerebral cortex, cerebral hemisphere, cerebrum we can call simply, that is the lateral ventricle. The diencephalon cavity is called as the third ventricle. So in a sagittal section, you would be able to actually demonstrate a third ventricle because it is a midline cavity which is situated in between the two thalamus of the either side. Okay, then the cavity of the midbrain, you are able to see a narrow cavity that is called as the cerebral aqueduct. And cavity of the hindbrain or rhombencephalon is your fourth ventricle, which is the cavity in between the pons, medulla anteriorly and the cerebellum posteriorly. So next, coming to the meninges of the brain. The meninges of the brain, they are actually the coverings of the brain which are actually protective in nature. Okay, so the meninges of the brain are coverings which are protective in nature, and the brain and the spinal cord are mainly covered by three coverings which are present as three layers from outwards to inwards. What you have is the Dura Mater, followed by that the middle layer is the Arachnoid Matter, and deeper to that what you have is the Pia Mater. Okay, so the Dura Mater is of compared to the other two coverings. It is very tough and made up of collagen fibers. Since due to its toughness, it is actually called as the Meninges. Whereas the Arachnoid Matter and the Pia Mater, they are a bit thin coverings and they are together called as the leptomeninges or leptomeninges singular. Okay, so external to the Dura Mater, there is an epidural space which is not seen in the cranial cavity, but in the vertebral canal where the spinal cord is present, there exists a epidural space. But here in the cranial cavity, the epidural space only you can see when there is a hematoma or collection of the blood. Then subdural space is below the Dura Mater, and below the arachnoid matter is the subarachnoid space, which consists of your cerebrospinal fluid. Same way, deep to the pia mater, what you have is the subpial space. Now, the existence of subpial space again is not accepted by everyone because there is some objection as to whether such space exists because the pia mater is very intimately attached to the brain tissue. Now, first coming to the Dura Mater. So again, with respect to the Dura Mater, in detail, kindly refer to the dural venous sinuses, dural folds and dural venous sinuses of the head and neck lecture series. Okay, so here I will just discuss about in briefly. Mainly the cranial dura is made up of two layers, the outer endosteal layer and the inner meningeal layer. So the endosteal layer is intimately adhered to the bone, Okay, the interior of the bone, it forms the endosteum, so inner layer covering of the bone. That is why it is actually called as the endosteal layer. Okay, now this endosteal layer along the sutures, it is continuous with the sutural ligament, so that is why very tightly adhered to the interior of the skull and also around the margins of the various foramens. Now, these two layers, they are almost together, but at certain places the meningeal layer separates from the endosteal layer and form a double layered fold which is called as the dural folds. So one dural fold which you are able to see in the sagittal in the mid sagittal fissure is the fox cerebri. So this is the fox cerebri which you are able to see here. Okay, so the other dural folds are fox cerebelli, tentorium cerebelli, and diaphragma cellae, and there is also one covering around the trigeminal ganglion which is called as the Meckel's cave. Okay, so these double layered folds consist within them the dural venous sinus. So the superior sagittal sinus and the inferior sagittal sinus which you are able to see. So like that there are various sinuses. You have straight sinus, right and left transverse sinus, sigmoid sinus, and more importantly the cavernous sinus. Okay, so as I told you, this endosteal layer is intimately attached to the bone. There is no epidural space in the skull, except when there is a fracture or hematoma is formed, epidural hematoma. Then this space is actually created because of the blood which is getting collected between the bone and the dura mater. Now, the Dura Mater, the main function, as I told you, it is a protective covering. It is made up of collagen fibers and osteoblasts are also seen in that. Okay, it is even though blood vessels are seen on the Dura Mater. So on top of the Dura Mater, mainly you have the middle meningeal artery, which supplies the greater part of the Dura Mater. Okay, so it is less vascular because as I told you it is made up of collagen fibers, not very much dependent for the nutrition, but the functions of these blood vessels mainly they supply the skull, interior of the skull, and the diploic tissue of the skull, because your skull has an inner and outer table of bone and inside you have the diploic vessels. It is a vascular tissue. Okay, so it also, when it dips into this mainly the large fissures, so the median sagittal fissure what I told you between the two cerebral hemispheres and also between the occipital lobe and the cerebellum, it intervenes as the tentorium cerebelli. Okay, so because of that what happens is it prevents the mechanical displacement during various movements, so it is protective in nature. Now the endosteal layer, as you trace, it is attached to the margins. This double layer you don't see below because along the foramen magnum the endosteal layer attaches. Okay, only the meningeal layer continues into the spinal cord. Now, how actually there is an epidural space present in the spinal vertebral canal is because the bones forming the vertebral canal, that is your bodies of the vertebra, have their own endosteal covering. Okay, so that is why the endosteal layer does not continues beyond, and between these and the meningeal layer there exists a epidural space. Okay, so this meningeal layer which continues beyond the foramen magnum and covers the spinal cord as the spinal dura. Okay, so that is the continuation, the thick fibrous coat. You are able to see the spinal dura and you are able to see the dural folds. The meningeal layer which forms a double layered fold is actually your fox cerebri and the tentorium cerebelli. So they are actually preventing the mechanical displacement of the brain and also they contain within them the dural venous sinuses. Okay, so this vertebral canal, the bones are again have their own endosteum, so there occurs a epidural space between the vertebral canal and the spinal cord. Okay, so in this epidural space lies your internal vertebral venous plexus. Okay, so the internal vertebral venous plexus is present. Now, the blood supply mainly to the dura has got many arteries, as I told you, mostly the middle meningeal artery with the frontal and the parietal trunk. Then branches of the anterior ethmoidal artery, ophthalmic arteries, and then the ascending pharyngeal branches, and also certain branches from the internal carotid arteries supply the dura. Okay, so mainly they supply the diploic tissue of the skull. That is what I told you. Okay, so middle, anterior, and posterior cranial fossa, the various arteries. The nerves are also mainly they are supplied by the nervus spinosum, again from the mandibular nerve, and then you have the maxillary nerve. Okay, and then posterior cranial fossa is supplied by the nerve from the branches of the ninth, 10th, and 12th nerve. Glossopharyngeal gives a meningeal branch. Okay, and your vagus gives a meningeal branch. And also branches from the trigeminal ganglion. And posterior part is also supplied by the upper cervical nerves. So that is why any inflammation of the meninges, the headache, in case of the anterior part of the meninges is involved, it is distributed through the trigeminal nerve. So you have frontal and parietal headache. But whereas if the posterior cranial fossa is involved, then pain is also referred to the neck because it is supplied by the cervical nerves, upper cervical nerves. So hematoma, you are able to see two types. One is the epidural. So only when there is a fracture of the skull, you are able to see an hematoma, which collects between your the bone, skull, and the dura mater. Now, epidural hematoma is mainly due to tearing of the arteries, which are superficial. Whereas subdural hematoma, not necessarily a fracture of the skull should occur. So internal rupture of the vessels might take place, some injury to the brain, and because of that what happens, a subdural hematoma is due to rupture of the cerebral veins, because veins are actually deeply situated rather than the arteries. So as I told you, meninges is inflammation. It might be due to so many things. So in meningitis, which affects the Pia Mater, is due to viral infections or bacterial or fungal infections. So one is most commonly the Japanese encephalitis, which affects the children most commonly. But nowadays vaccines are available for these against this viral encephalitis. So bacterial encephalitis, even from a pneumococcal infection, meningitis might occur. So from the middle ear, infections can spread through the roof of the middle ear. From the uh, what do you call, the roof of the nasal cavity, these veins have a direct connection with the CSF. They drain into the CSF. So naturally infections from there can enter, and even infections from your dangerous area of face can enter into the cavernous sinus. Okay, so these are all the spread of route of infections. So symptom includes fever followed by headache, then you have the neck stiffness and vomiting and fatigue. Now, coming to the next layer is the arachnoid matter. So, it is deep to the dura mater. What you are able to see is the arachnoid matter. Okay, and from the deeper surface of the arachnoid matter, you can see some trabeculated tissue which is running in the subarachnoid space, okay, and attaching to the Pia Mater. So because of this trabeculated appearance, which appears like a cobweb, it is labeled or named as the Arachnoid Matter, Arachnidae, where the spider, the phylum. Okay, these are the arachnids. Okay, so trabeculated tissue which divides the subarachnoid space into numerous trabeculated spaces. So again, this is avascular, and not only that, it is also insensitive to pain. Okay, this arachnoid matter, it is made up of 5 to 6 layers of cells. Even though it is a thin membrane, it is made up of 5 to 6 layers of cells which are held together by the tight junctions and desmosomes. Because what happens is this CSF should not have any contact with the subdural space where the numerous arteries are present. So that is why it is 5 to 6 layers in thickness. And as I told you, the CSF is present in the subarachnoid space, okay, which is what you call the subarachnoid space consists of the cerebrospinal fluid. So the arachnoid is mostly adherent not to the dura mater, which is opposed against it, but more towards the pia mater. Okay, so there exists definitely a very much appreciable subdural space between the dura mater and the arachnoid mater. Okay, so in the spinal cord, what happens is the subarachnoid space, which is present deep to the arachnoid mater, and at the end, what you are able to see where the arachnoid ends as the phylum terminale. So there is a free flow of the CSF between the coverings and the lower or end of the spinal cord, which we call it as the conus medullaris. So this free flow of CSF is also called as the fifth or the terminal ventricle. Okay, so the arachnoid matter is continuous with the epineurium of the peripheral nerves. The peripheral nerves, it is continuous with the epineurium. And remember, certain veins drain into the CSF directly to the roof of your nasal cavity. Then this arachnoid matter is also continuous around the optic nerve. So mainly the dura mater, the endosteal layer forms the periosteum of the orbit. The meningeal layer is continuous with the sclera. Then the optic nerve deep to it, what happens is it is continuous around the arachnoid matter. Because of that, any increase in the intracranial pressure when there is a defect in the drainage or circulation of the CSF, it is very much reflected in your eye. You will be able to see the pupil edema. Okay, so the edema gets, the pupil gets bulged out. Okay, you can make out that. The arachnoid, in certain places, they have finger-like projections which are called as the arachnoid villi. Mainly you see it in the superior sagittal sinus. So finger-like projections of the arachnoid matter, which is called as the arachnoid villi. These villi, when they aggregate together, they form the arachnoid granulations. Okay, so the arachnoid granulations, through which what happens is the CSF is actually drained into your venous blood. CSF is drained into your venous blood, that is into the dural venous sinuses. So you can see in aged skulls, these granulations, they produce a very much markings on the interior of your vault of the skull, like a depression or pits. You can see when you see the pits very marked and deep, that means it is a very aged skull, the age advanced in elderly people. Okay, so the subarachnoid space deep to the arachnoid matter mainly consists of CSF, and the blood vessels are actually traversing through it. At certain places, what happens is the CSF extends around the optic nerve, as I told you, large collection of CSF. You are able to see some large collections of CSF are seen between the arachnoid and pia mater. So the arachnoid and pia mater are adherent together. It is arachnoid is opposed against the dura, but in close contact with the pia. But at certain places, when you look, the arachnoid and pia, like the meningeal and the endosteal layer of the dura mater, they separate out and it is filled with CSF. Large pools of CSF are present and they are called as the cisterns of the brain. If you look carefully, these cisterns of the brain are mainly seen along the base of the brain. Okay, and these cisterns mainly contain the blood vessels. So the blood vessels which are mainly entering through the base of the brain, whether the carotid system of arteries, internal carotid artery enters through the carotid canal, and the vertebral arteries enter via the foramen magnum. Both are present in the base of the skull. So through the base of the brain, when they enter, they are bathed in this CSF. Okay, so the cisterns which are present around in the brain, mainly one is the largest cistern, cisterna magna, is the cerebellomedullary cistern. So here we are able to see the cerebellomedullary cistern below the cerebellum and behind the medulla oblongata. And above you have the roof of the fourth ventricle. This is your fourth ventricle. Okay, so you have the cerebellomedullary cistern. Okay, so mainly it consists of some olivary veins, posterior olivary and lateral medullary veins. This cistern is continuous above with the lateral ventricle. So the openings in the roof of the fourth ventricle, sorry, I told it as lateral ventricle, from the foramen of Magendie, an opening in the roof of the fourth ventricle, the CSF escapes into the subarachnoid space. Okay, because until then the CSF circulates only within the ventricles, lateral ventricle, third ventricle, cerebral aqueduct, and fourth ventricle. The CSF within the ventricular system escapes out of this ventricular system and enters the cerebrospinal fluid. And one place is the cisterna medullary, cerebellomedullary cistern or cisterna magna, which you are able to see here. The next cistern, what you see is the cisterna pontus. You are able to see in front of the pons, and that is continuous with the CSF of the spinal cord. Okay, so the subarachnoid space of the spinal cord. Above, it is continuous with one more cistern, cisterna pontis. Okay, cisterna pontis is continuous above with the interpeduncular cistern. So that is the interpeduncular cistern. It is continuous above with the internal peduncular cistern, posteriorly along with the cisterna magna. Okay, so the main content is your basilar and inferior cerebellar arteries. Okay, the basilar artery, which is the terminal division of the vertebral artery, union of the two vertebral arteries to form the basilar artery. And these are branches from the vertebral arteries. Then, as I told you, interpeduncular cistern, which is situated between the two cerebral peduncles, communicates with the pontine cistern, and one more cistern is the cisterna ambiens, which you will see here. And anteriorly and rostrally it is also continuous with the chiasmatic cistern, which is situated near the optic chiasma. So consists the main arterial circle, anastomotic circle, the circle of Willis. Then you have the cisterna ambiens around the base. It communicates with the interpeduncular cistern and quadrigeminal cistern. So the quadrigeminal cistern you see here. So around the midbrain, base of the midbrain, you come across this cisterna ambiens, and it mainly consists of the great cerebral vein of Galen. So you're able to see the subarachnoid space with its trabeculated tissue and how the blood vessels are situated. So interpeduncular cistern mainly consists of the circle of the Willis, which you are able to see here. Okay. So the great cerebral vein of Galen is the content of the cisterna ambiens. Then you also have one cistern which is called as the Silvian System, which consists of your middle cerebral artery along the Silvian fissure or lateral sulcus of the cerebrum. So don't worry about what is lateral sulcus. All this we will be seeing in the succeeding classes. So mainly consists of the middle cerebral artery and superficial and middle cerebral veins. So one applied aspects is the cisternal puncture, which is carried out in the cisterna magna, cisterna cerebellomedullary cistern to withdrawal of CSF. So not always advised. If the lumbar puncture which is done at the L3 L4 level is not possible, then actually cisternal puncture has to be applied or performed because it is a bit dangerous procedure. Complications are hemorrhage and damage to brain tissue, or sometimes it might be fatal because here the medulla, the respiratory and the cardiac centers are present. So sometimes if it is injures that area, then respiratory failure will occur. Okay, so it is mainly done to the suboccipital region. To the needle is introduced via the suboccipital region to withdraw CSF. Not commonly advised, only if there is no other choice. If the lumbar puncture if not possible or fails, then you go for this cisternal puncture. So the next covering, the innermost covering is the Pia Mater. The Pia Mater is again a thin avascular membrane closely related to the surface of the brain, and it also runs along all the gyri and sulci. Deeper into the sulci it covers, and blood vessels are present on the surface. Not only on the surface, deep to it also the blood vessels are present. So there is a covering of Pia Mater around these blood vessels, and that forms the perivascular space. So sheath of Pia Mater around the blood vessels, and that is actually called as the perivascular space, compensating for the lymphatic system. Okay, so mainly they compensate for the lymphatic system. Now again, this Pia Mater is 2 to 3 layers in thickness, and they are held together by the gap junctions, and that should prevent the CSF communicating with the blood vessels which are present deeper to the Pia Mater. Okay, so two layers, one is the epipia and other one is the pia glia limitans. So here are able to see the Pia Mater is made up of epipia, and then the pia glia limitans, which is mainly formed by the limiting glial membrane by the foot plate of these astrocytes. So they are responsible for forming the blood-brain barrier, so that the blood vessels and the brain tissue, they don't come in contact directly. The blood-brain barrier is actually formed by the vascular endothelium and also by the vascular foot plate. Here it forms a continuous membrane, and the Pia Mater also performs the function of pinocytosis. Some small molecules it can ingest, and it also absorbs certain neurotransmitters which is seen on the surface of the brain. Okay, so in the region of the ventricles, you are able to see the Pia Mater forms a double layered fold. It projects into these ventricles as double layered fold, which is called as the Tira choroidea. So within this Dura choroidea, you have this vascular tissue called as the choroid plexus. So the choroid plexus is seen even lateral ventricle, and mostly you are able to see in the fourth ventricle and also in the third ventricle. Okay, so these choroid plexus, they are actually uh, capillary plexus. Okay, which are again lined by the ependymal cells. They are actually the choroidal ependymal cells. Ependymal cells are again 2 to 3 types you have, mainly the choroidal ependymal cells which lines the choroid plexus. Okay, so these are mainly responsible for the filtering the CSF from these choroid capillaries, which forms the choroid plexus. Okay, so from the blood what happens is the CSF is actually filtered via the choroidal ependymal cells. Okay, so they are called as the choroid epithelial cells which secretes the CSF. So here are able to see CSF from the choroid plexus and the ependymal cells lining it in the lateral ventricle. So from there the CSF to the third ventricle, cerebral aqueduct, the fourth ventricle. The roof of the fourth ventricle has three foramina, one median foramina called as foramina of Magendie and two lateral foramina called as the foramina of Luschka, through which the CSF enters into the subarachnoid space. So here you are able to see the cisterns, cisterna, This is actually your magna, then pontine cistern, then interpeduncular cistern, chiasmatic cistern. So here you have the cisterna ambiens, and then you have the quadrigeminal cistern, and then you have the lateral cisterns. So they're all you can see. Okay, from the fourth ventricle, uh, you're able to see it is escaping into the subarachnoid space. Fourth ventricle to subarachnoid space. Then, after circulating through the arachnoid villi, which we have seen in the superior sagittal sinus, it enters into the venous blood. So, ultimately, venous blood has to enter into the heart. Okay. So, from there, what happens? Again, the arterial blood through the internal and vertebral system of arteries enters through these ventricles through the choroid plexus. So that is how the CSF is secreted, circulated, absorbed, and it is a cycle. Okay, so with this, I have completed the introductory class on the brain or neuroanatomy and also the meninges. So the next class we will be starting with the brainstem. So thank you for your patient listening.